Thank you for coming to today's tutorial. I'm Carl Peters, and on behalf of BMG Labtech, I'm happy to welcome you to today's tutorial and to introduce Jacob Ayers. Jacob is going to tell us a little bit about some of the work his group is doing to develop assays with the PHERAstar FSX that will help to discover prion treatments. At BMG Labtech, we take pride in the fact that we are very responsive to the needs of the scientific community. We are able to be so responsive because we are focused solely on the production of microplate readers. BMG's commitment to excellence is exemplified by the PHERAstar FSX, our flagship HTS reader. The PHERAstar FSX is the fourth in a line of HTS readers that has developed over time to be able to perform multiple different detection modes as listed on the right-hand side, and to do all of these with the highest speed and sensitivity. It can do this through the use of assay-specific modules for defined assays, a spectrometer for absorbance for selecting wavelengths from 220 to 1000 nanometers. The speed is aided by the fact that there are two pairs of matched PMTs that allow for simultaneous dual emission. And for advanced assays, you can select laser excitation sources. As today's presentation by Dr. Ayers will feature the use of time-resolved FRET, I would like to point out a few of the things that the PHERAstar FSX has that are advantages for detection in this mode. The first is, of course, the optic module system, which is depicted on the right-hand side of the screen. And as I previously mentioned, this enables for simultaneous dual emission. And you can see that really within this optic module, you have all of the filters, dichroics, beam splitters, everything you need to perform these assays and to perform simultaneous dual emission quickly. The FSX can be equipped with a time-resolved fluorescence laser, a UV laser for excitation, and it also has dedicated matched detectors to improve for sensitivity. When you consider a high throughput screening instrument, your primary concern is going to be with the ability to perform assays in a miniaturized format. Therefore, I'm only showing you the 1536 well LOD for the PHERAstar FSX, and you can see in this comparison that there is no reader more sensitive. Without further ado, I would like to introduce Dr. Jacob Ayers. Dr. Ayers has long been interested in prion diseases. After getting his undergraduate degree from the University of New Hampshire, he studied prion diseases under the mentorship of Dr. Jason Bartz at Creighton. There, he investigated the mechanisms that govern prion strain targeting using hamster-adapted prion strains. He then joined the laboratory of Dr. David Borchelt at the University of Florida and began studying the properties of ALS-linked protein superoxide dismutase 1. He then joined the faculty at University of Florida as a research assistant professor and continued his studies in ALS and began investigating induction of Parkinson's-linked protein alpha-synuclein in a transgenic mouse model. Dr. Ayers then joined the Institute for Neurodegenerative Diseases, IND, at UCSF in 2018 and is currently the lead biologist for in vitro drug discovery. His work focuses on the discovery of therapeutic interventions to treat and prevent the progression of neurodegenerative diseases. Today, he will tell us a little bit about how they use the PHERAstar FSX to further this work. Dr. Ayers?
So first, I'd just like to say thank you to BMG for the opportunity to share some of our recent data that we've generated at the IND here at UCSF. So today I'm going to be discussing some new assays that we've developed in our lab to help out our drug discovery efforts in discovering small molecules to treat several neurodegenerative diseases. And specifically today, I'm going to be concentrating on assays that we've developed to detect tau prions, or those proteins that accumulate in the brains of Alzheimer's disease and several other tauopathies. So prion diseases encompass a number of neurodegenerative diseases. So initially, they were made up of just one disease called spongiform encephalopathy, which makes up mad cow disease and Creutzfeldt-Jakob disease. And it was shown early on that these diseases were caused by a single cellular form of the prion protein, which, when misfolded, becomes toxic and accumulates within the CNS of patients and animals. But over time, a lot of similarities of some of these other neurodegenerative diseases were shown to be very similar to prion diseases. And it's now understood that these other diseases, such as Alzheimer's disease, Parkinson's, ALS, and Huntington's, are also comprised of proteins that misfold and can form prions. And so these include amyloid beta and tau in cases of Alzheimer's disease, alpha-synuclein for Parkinson's disease, SOD1 for ALS, and a lot of others. So the interesting thing about these infectious agents, unlike bacteria or viruses, is that they contain no nucleic acid. So what happens is that a normally folded cellular protein becomes misfolded and toxic. And so you get conversion of the normally folded protein into its toxic form by what's called templated misfolding. And these misfolded proteins then aggregate and thereby exert their, what some believe are the gain of toxic features of these proteins. So the way that prions are able to propagate and accumulate within the central nervous system is, like I mentioned, the misfolded toxic form is able to template its conformation onto these normal, non-toxic cellular forms of the protein. And what happens is that this kind of becomes a domino effect, and that you have more of the normal protein that becomes conformationally misfolded. It then accumulates, and what happens is that you get these large aggregates and these fibrils that form within the central nervous system. And when these break apart, they actually form more seeds and more area for this normal cellular form of the protein to bind and then convert. And so this formation and the fragmentation of these aggregates actually allow for the continued propagation of these proteins. And this has been shown for a while that a lot of these proteins, like amyloid beta, very similar to the prion protein, forms aggregates. And like I mentioned, this happens for amyloid beta, for tau, for alpha-synuclein, TDP-43 FUS. More and more proteins that are implicated in neurodegenerative diseases are showing to have these very similar properties. So these proteins, once they become misfolded, tend to clump up and form these aggregates, which you can see in these immunohistochemistry pictures here. And what's also noticed is that these aggregates actually are able to move throughout the central nervous system in what some suggest might be through neuroanatomical transport, and that from anatomically connected regions of the brain, these aggregates can actually spread. And so it might start out in one area of the brain, but over time, these proteins and these aggregates actually accumulate in synaptically connected areas of the brain. And this has been shown, again, through a lot of neurodegenerative diseases, through tau and amyloid beta in Alzheimer's disease, in alpha-synuclein for Parkinson's disease, and in some other proteins like TDP-43 for ALS. So there have been a number of mechanisms that have been put forward to explain how the aggregated proteins are spreading throughout the central nervous system. So a couple methods that could explain local spread are that these toxic proteins Once inside a cell, the cell will die, spilling out its contents of these aggregated proteins, which can then be taken up by nearby cells. Also, what can happen are these cells can actively be the aggregates can be actively transported out of the cells through a method like exocytosis, and then be taken up by nearby cells. And what's also been shown, like I mentioned, is this network spread, so that a neuron can actually shuttle these aggregates down its axon, and through transsynaptic spread, it can actually push these aggregates to synaptically connected cells. So, as I mentioned before, tau is a component of some of these aggregated proteins in diseases such as Alzheimer's disease and several other diseases which are within a family called tauopathies. And so tau it's a structural protein whose normal function is to stabilize microtubules, which are really the structural support within the axons of neurons. And so what happens in tauopathies and in Alzheimer's disease is that tau, like I mentioned, it aggregates within neurons and forms what's called neurofibrillary tangles. And you can see a immunohistochemistry picture here with the brown deposits here being aggregated tau in the form of a neurofibrillary tangle. And so what happens is, again, the soluble form of tau becomes misfolded through templated conversion and then aggregates to form these structures. And so just a little more information about tau. So the gene for tau actually expresses 6 different isoforms of the protein. And these are differentiated based on these several domains within the protein. So you have these N-terminal domains, 2 of them, and then these 4 repeat domains near the C-terminal of the protein. And so this is really important when you're looking at these different diseases because not all of these isoforms actually accumulate within these diseases. So there are several diseases in which just the 4R isoforms, these bottom 3 isoforms, accumulate into tau aggregates within the disease. And these include several tauopathies such as PSP, CBD, and others. And then there's another form of tauopathy called Pick's disease, in which only the 3R forms of tau are found within the aggregates. And then interestingly, there's also other diseases like Alzheimer's disease and chronic traumatic encephalopathy, in which you have all 6 isoforms of the protein accumulating. So both the 3R and the 4R isoforms. So recently, a lot of work's come out of Michel Godert's lab that have looked at the actual conformations of the tau protein that accumulates in these different tauopathies. And so, like I mentioned before, when you have this aggregated tau, what happens is that these actually form fibrils, which are these really ordered structures of tau that accumulate within the neurons in the brain. And so what they've been able to do is, by using cryo-electron microscopy, is actually show that the aggregates of tau within these different diseases actually are comprised of different folds of the tau protein. And so this is really important moving forward for drug discovery, in that we're not just targeting one conformation of tau, but you see for these different diseases like CBD, Pick's, Alzheimer's, and CTE, that the tau protein is actually a little bit conformationally distinct in each of these disorders. And so what we've done over the last couple of years is to build on some initial experiments that were done in Mark Diamond's lab in order to have in vitro assays that allow us to detect prion activity from different sources. So what this entails is having HEK cell lines that overexpress different forms of tau, which are fused to yellow fluorescent protein, allowing us to directly visualize the protein within cells. And in some of our initial experiments that were done by Amanda Werman and published back in 2016 is to express a certain 4R form of tau with a couple different mutations. And what we found is that when you take brain homogenates from postmortem tauopathy patients and add these to the YFP tau expressing cells, is that some of the patient material actually induced aggregates to form within the cells. And so you see that when tau aggregates within these cells, that it forms these really bright puncta. And these are very similar to the same aggregates that are formed within these particular patients. So what was interesting in these first experiments that we found is that there's actually some specificity to the protein that's being expressed. So if you're expressing 4R tau, only those 4R tauopathies are able to induce aggregation. So Pick's disease is a 3R tauopathy. Alzheimer's and CTE are 3R and 4R tauopathies. So you can see when you directly measure the amount of aggregation within these cells that only these diseases that 4R tau accumulates and aggregates, you can see detection of aggregates within these cells. Now, Amanda also made another cell line that expressed both 3R and 4R tau, which both had a yellow fluorescent protein tag on them, so we could visualize these. And what she found is that now, if you add Pick's disease, which I mentioned is a 3R tauopathy, you can get induction of these aggregates. And also with Alzheimer's disease and CTE, which are 3R and 4R tauopathies. Now, again, you can get induction at pathology, showing that there's some specificity to the tau prions that are in the homogenates of these patients. They're only able to induce the cells that have the correct isoforms being expressed. So although these tau YFP cellular assays I've been discussing are very powerful tools in order to measure the prion activity within patient material, there are several complications that arise due to having that yellow fluorescent protein tag on the end of tau. So you can see here, just for your perspective, that the tau that we're expressing here is about 384 amino acids long, whereas the yellow fluorescent protein is 238. So you have this big bulky tag, which is over half the size of tau itself. And this could really impact the folding of tau. And we know in my previous slides mentioned how important the conformation of tau is for these various diseases. So we don't want anything to impact the potential folding or misfolding of tau. One of the other impacts that this could have is for drug discovery. So we're trying to develop small molecules to bind and inhibit the aggregation of tau. We don't want any influence from the yellow fluorescent protein or any tag on tau to impact that potential interaction. So to get around these problems, what we've done is actually developed a tag-free aggregation assay. So very similar to our in vitro cell assay, we've developed stable cell lines that just express untagged versions of tau. And all these were made in HEK293 T cells. And so the problem here is that without the YFP tag, we need to come up with another means to measure the aggregation of tau or the formation of these fibrils that occur within the cell. So we turn to HTRF means in order to measure the aggregation fibril formation within the cells. So the first thing that we had to do was obviously make stable cell lines that express untagged tau. So we used, again, HEK293 T cells to express different versions of tau. You can see the Western blot of several clones that we were able to produce. Here you see our normal tau YFP cell line. You see this is shifted up due to the yellow fluorescent protein tag, that bulky YFP tag. And here you see the different clones of untagged tau that we were able to produce. So we had several clones that had high expression of tau. So the next thing we had to do was to ensure that they were actually making and capable of producing aggregates. So first off, we just wanted to look at the immunoreactivity of several different tau antibodies in our regular tau YFP expressing cells. And so we used different commercially available tau antibodies, the AT8, the MC1, and the PHF20 antibody. And you can see here just the YFP coming from the fluorescent tag on the protein. And then when we used a secondary antibody to these primary antibodies, you can see without induction of pathology that the AT8 and MC1 antibody didn't show too much immunoreactivity. The PHF20 seemed to label some of the soluble tau that was around in these cells. However, when you induce aggregation within these cells, again, you have the fluorescence coming from the tag on the tau YFP cells. And now, when you stain with either AT8 or MC1, you see strong localization of these antibodies to the aggregates within the cells. Again, you have a lot of soluble immunoreactivity with this PHF20 antibody. So now moving to our untagged cells, you can see that without the addition of tau prions, in which there'll be no, or there shouldn't be any accumulation of tau aggregation, again, you had some immunoreactivity to the AT8 antibody, nothing with MC1, and again, you see plenty of soluble tau being expressed within this particular cell line. However, now when you add tau prions into this line, we're able to determine, just using these antibodies, that we are getting induction of tau pathology. So again, you see the aggregates with the AT8 antibody, the MC1 antibody, and the soluble and aggregated localization of tau with the PHF2 antibody. So now that we knew that we had a cell line that expressed a tagless version of tau, and through immunocytochemistry we showed that we can induce aggregation by adding tau prions to these cells, we needed a system that we could miniaturize and also detect the accumulation of tau aggregates within the cells. So for this, we turned to HTRF, or homogeneous time resolved fluorescence. And SysBio offers a number of kits available to detect aggregated proteins, including tau, and in the example here, alpha-synuclein. And what the technology is by taking antibodies to a similar epitope, they're able to fluorescently label them with a donor and an acceptor fluorophore. And by mixing, say, a lysate that contains aggregated synuclein or tau, you can add these antibodies. And if the antibodies are close enough, you get a FRET reaction. So you get a fluorescence energy transfer from the donor to the acceptor. And you can use a plate reader like BMG's PHERAstar FSX to read the fluorescence coming off the acceptor. So how we first went about looking at this is to find out how well it correlated with our existing tau YFP assay. So in this assay, what we're able to do is, in a 384 well plate, we're able to add tau prions to, of course, the tau YFP cells, and we let this incubate for about 3 or 4 days in order to allow time for the induction of aggregation. And this is read out on a automated confocal microscope. So we take multiple pictures of each of these wells, and then we can quantify the aggregates based on the YFP fluorescence. And you can see here that with an increased amount of tau prion being added to these seeds, that you get a nice dose response or an increase in aggregation that occurs with an increased amount of tau prion being infected. So we also wanted to see how this correlated, of course, to our tagless tau cell line. So again, we added tau prions to our cells in a 384 well plate. And then instead of reading the cells due to fluorescence, what we did was actually lyse the cells within the plate and added antibodies to these lysates to detect it. And then we read this on BMG's PHERAstar FSX. And what we found is a very strong correlation to what we observed in the tau YFP cells, is that when you add increasing amounts of tau prions, you get an increased amount of HTRF signal, which in this case is tau aggregates within these cells. So of course, in our lab, we're interested in developing compounds that inhibit this aggregation. So we also want to make sure that we can detect the decrease in aggregation within these cells in, say, an HTS screen. So again, we add tau prions to our tau YFP cells. We can add certain inhibitors that we know will inhibit aggregation. And what we see is that when we read the YFP signal, when we add this compound with increasing doses, you get a nice dose response curve. Now with our tau or the tau untagged cells, you add the same tau prions, the same compound. Now by reading the HTRF signal, again, we get a very nice dose response being read with the PHERAstar FSX, showing that you have a very nice correlation between our YFP assay and our tagless YFP cells. So again, another application that we can use this HTRF is actually to detect prions straight from the brains of some of our transgenic animals and from the brains of some of the post-mortem patient material that we have. So in this instance, we used a brain from a diseased tau transgenic mouse. So at late disease, these mice accumulate lots of tau prions within their brain. We're able to homogenize that, and in a 96 or 384 well plate, we're able to add the HTRF antibodies. And what we can see is that when you look at the tau transgenic mouse brain, which is in orange, or a non-transgenic mouse, we did dilutions of these two homogenates, and of course you see with looking at the HTRF signal that you have an increased amount of tau aggregates that are detected in the brain. So this just shows you again that you can use this technology to detect tau aggregates straight from patient or mouse tissue as well. So in summary, the HTRF has really given us the ability to measure tau prion levels both in our cell models and in brain tissue. And this is important because we're able to see this independent of fluorescent tags. So what this enables is for miniaturization of these assays and potentially for doing HTS assays for our compounds that we're screening. And with that miniaturization, of course, we're able to develop high throughput screens in order to find inhibitors for some of these prion aggregates. And this is really important using a tag-free system instead of having a big bulky YFP tag on the end of this, which of course could impact the folding or the binding of some of these compounds to the protein itself. So with that, I'll wrap up, but I just want to really thank all the members of the IND that have helped out with this research, especially our director, Stan Prusner. So I thank everyone for your attention, and I'll be happy to answer any questions. Thank you.
Thank you, Dr. Ayers. It is gratifying to know that BMG Labtech plays some small role in the important work that you are doing. In closing, thank you for your attention and have a great day.